Welcome to Part 2 of Lesson 3.2, How Do Normal Cells Become Cancer Cells? We pick up from the first part of the lesson by engaging in a do now. The key points are that proto-oncogenes normally stimulate proliferation and tumor suppressors normally inhibit it. Mutations to both proto-oncogenes and tumor suppressor genes are the first steps in cell transformation. We begin the do now by considering what types of mutations are seen in cancer cells. By now we have learned about mutations happening through transposons, chromosomal rearrangements, and changes to individual nucleotides. Any of these can adversely affect two major types of genes associated with cancer, proto-oncogenes and tumor suppressor genes. We focus on SNPs because these are some of the most common mutations present, and we know of some SNPs that cause inherited cancer. We have learned the concept of synonymous and non-synonymous mutations to protein coding regions. But many more SNPs will be found in non-coding regions, simply because non-coding regions form the largest part of the genome. In the lesson's activity, students will use the UCSC Genome Browser to access the online Mendelian Inheritance in Man and flagged SNP databases that link DNA mutations to cancer. They will then look at the types of mutations that occur in 7 genes associated with cancer. They will learn how these SNPs in these genes are associated with cancer and predict types of mutations common in other proto-oncogenes and tumor suppressor genes based upon gene function. The key points students should take away from the activity are that only non-synonymous SNPs in an exon affect protein structure directly, but SNPs in introns might affect protein structure indirectly if they change how splicing occurs. The key points of the wrap-up are that mutations to proto-oncogenes and tumor suppressors are the first step in cell transformation. However, full transformation requires a third mutation in genes regulating aging and death. Fully transformed cells are both hyperproliferative and immortal. To wrap up the activity, show the final table from the worksheet. Next, animate the slide to show how turning tumor suppressor genes off prevents both cell cycle arrest and apoptosis, allowing damaged DNA to replicate. However, proto-oncogenes need to be mutated to allow the cell to hyperproliferate. Transformation begins when cells with damaged DNA continue to proliferate because both tumor suppressor genes and proto-oncogenes are mutated. Hyperproliferation is the first step towards transformation, but transformation is only complete when the hyperproliferating cell becomes immortal. This requires a mutation in a third type of gene. We will examine this type of mutation in Lesson 3.4. For homework, have students read the workbook for Lesson 3.2 and answer the homework questions. One of the most common questions students ask is, can RNA or proteins be mutated even if the corresponding DNA is not? First of all, the term mutation only refers to DNA, but proteins and RNA can develop sequence errors as they are being made. However, this will only have a short-term effect because RNA and protein are frequently replaced. Long-term effects to a cell only happen if the DNA in that cell is mutated. So all the progeny of that cell will also inherit this mutation. Another question students ask is, what is the difference between proto-oncogenes and oncogenes? All normal cells express proto-oncogenes, and they are critical for normal cell function. When a proto-oncogene is mutated and loses its normal function, then it is called an oncogene. At the end of the lesson, collect student worksheets to assess whether students can describe how mutations in proto-oncogenes and tumor suppressors lead to cancer. We will continue our investigation of cell transformation in future lessons with an examination of how transformed cells become immortal. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.